Okay, then I will introduce our second speaker, Megan Bonsor. She is a PhD student at the Max Delbruck Center in Berlin, and her poster is entitled Investigating the Multi-Omic Impact of Huntingtin Aggregates on the Melanogaster Brain. And the poster number is A15. And very welcome. You hiding there. Very welcome.
Okay, thank you everyone for coming today. Hopefully you can see me over the screen. So today I'm going to be speaking about my research project, if I can get this working. Great. Entitled Investigating the Impact of Huntingtin Aggregates on the Drosophila melanogaster Brain. So to introduce my project to you, I have to introduce you to the concept of aggregation in Huntington's disease, something that many people today have already spoken about, so please bear with me. So in this project, we specifically use Huntingtin exon 1. And Huntingtin exon 1 is an N-terminal protein fragment consisting of the poly-Q domain, and it's been found in vitro and in patients to form aggregates and inclusion bodies. And it's hypothesized that possibly these aggregates and inclusion bodies play a role in impairing the proteostasis network of the cell. And in turn, maybe it's this impairment of proteostasis that could be contributing to this vast neurodegeneration that we see in Huntington's disease brains. However, the link between aggregation and neurodegeneration is not so clear-cut. For example, some of the neurons that degenerate the fastest and the most severely, such as the medium spiny neurons, do not necessarily have the most amount of Huntingtin exon 1 aggregates. So therefore, there's quite a lot of unanswered questions regarding aggregation in Huntington's disease. And the two questions outlining my research specifically is what mediates this selective neuronal vulnerability that we see in Huntington's disease? Why are some neurons degenerating more severely, more aggressively than others? And what role does Huntingtin exon 1 aggregation specifically play in Huntington's disease? What is it these aggregates are doing on a cellular level, transcriptomically and proteomically? So to address these questions, we've established a brand new in vivo model for Huntingtin exon 1 aggregation. And this is all based in the fruit fly, the Drosophila melanogaster. So our fruit flies panneuronally co-express a Huntingtin exon 1 fusion protein, which is fused to a fluorescent molecule, M-neon green. And they also co-express a Huntingtin exon 1 tagged with a fluorescent molecule called M-scarlet. These fusion proteins contain either Q75, so 75 glutamine repeats, or 17 glutamine repeats, Q17. And due to the overlapping emission and excitation spectra of the M-neon green and M-scarlet fluorescent molecules, when our fusion proteins co-aggregate together, there's a distance-dependent energy transfer, also known as FRET. And it's this FRET, this emission of a higher wavelength of light, that allows us to look at the aggregates in an in situ context, so within the brain of the fly. And our first step in making sure that this model was sound was first to look at, well, is our fusion proteins, are they aggregating? And secondly, can we detect these aggregates using FRET like we hope we can? And our first step in doing this was to approach it from a confocal microscopy point of view. And we could confirm that in our Q75 expressing flies, we saw a FRET-positive fluorescent puncture. Conversely, in our Q17 expressing flies, this is the brain of the fly, we did not see these fluorescent FRET-positive puncture. So this was kind of giving us the idea that maybe our proteins are aggregating as we hoped. And to confirm the presence of these Huntingtin SDS-stable aggregates, we use an anti-Huntingtin antibody called PHP2. And we use this to try and immunoprecipitate these supposed aggregates from our fly samples. And through analyzing these results using a gel as pictured here, we could see that in our Q75 expressing flies, there were SDS stable structures which are sequestered at the top of the gel. So this kind of confirmed that we do have SDS stable aggregates in our Q75 expressing flies. What's interesting and kind of reassuring as well for our model is that these flies expressing Q75 had a significantly reduced lifespan, as you would expect for a Huntington's disease model. And finally, we were able to isolate single cells from the whole brain of the Drosophila, and these were live single cells. By analyzing these single cells using a method called FACS, we were able to show that there's an increase in a FRET-positive cell population with our Q75 expressing flies. And we can use a gating strategy of this FACS to select cells that have this FRET-positive signal, and therefore select cells, live cells, that have aggregates. Looking to the future, we currently have 2 different samples put forward for mass spec. We have proteomic samples using whole brain Drosophila samples, and we also have immunoprecipitated aggregate samples put forward for mass spec to try and analyze the aggregate's effect on the proteome specifically. And secondly, we've been able to use our FACS method that I just spoke about to enrich for cells containing these FRET-positive aggregates. And we've put forward a data set for single-cell sequencing to really interrogate what cells have aggregates and how are these cells transcriptomically different to cells that do not contain aggregates. Eventually, we're hoping to combine this proteomic and this transcriptomic data sets in order to functionally validate protein hits and gene hits using our Drosophila model. And with that, I'd like to just thank all of my people who have made this project possible, and I'll be happy to answer any questions you may have.
Thank you so much. Do we have questions from the audience? While we are waiting, yes, there is a hand up there. So then you will have a question from the audience immediately.
So it's a basic question. Nice work. How do you select the flies which has this mutant Huntingtin? Like, in the sense you already get it, or like you do a cross and all, right?
Yes.
You should select the flies which have this Huntingtin, right? Mutant.
Yes.
So how do you do that? It's based on FRET or something else?
No, so when we cross the flies, I wish it was based on FRET, good question, but no, we use a standard crossing scheme where we use homozygous male flies that express our Huntingtin under the control of the UAS promoter, and then we cross them using balancer phenotypes. We can tell which ones are homozygous and which ones are heterozygous, and then we only cross the homozygous males with our ELAV, so our neuronal driving GAL4 line, which enables the expression via UAS. It's a more Drosophila heavy topic, so if anyone wants to know more, please come to my poster, but hopefully that answers your question. Yes, yes, yes.
There we have another question. Oh, several questions.
Hi, thank you very much. That was very nice. So I was wondering, since your final aim is actually to look into cell-specific vulnerability, so I guess you'll want to look to MSN and some other systems. So I was wondering whether you know whether you can translate this tool into a mammalian system of some sort.
Yes, definitely. So the flies are obviously a jumping off point, but the flies actually have a lot of homologous neurons to humans, more than you would probably think. So there is a homologous region that's similar to the striatum. So I think our first port of call, once we get our single cell data set and see what cells are kind of different to others, we can look for cell-specific drivers in Drosophila, which is way quicker than using another model. And we can probably up and, you know, overexpress certain modifiers and things. Obviously, everyone has their favorite model, right? But ultimately, we would do initial tests in Drosophila, and then hopefully it's definitely translatable to other models, yes.
So did we have another question there? Okay, with that, thank you very much. This was really fascinating. I'm thinking about starting with flies instead of mice now.
Ooh, do it. Thank you.